It's 10 minutes stalling, isn't it?
Yeah, but I'll speak to them first.
Yeah, cool. Yes, should be like that.
Okay, everyone.
Just test the mic. Austin, can you hear me? Yeah,
If you can hear me, put W in the chat. If you can hear me, guys, can you put W in the chat so I know? Yes, excellent, excellent. Smashed it. Okay, guys, welcome. Welcome to all of you students, to all of you hard-working students who've been working hard for the past 2 years for this season. This is the exam season of 2024. Okay? Today, we're going to be going through Biology Paper 1, predicted topics for your exam tomorrow. Now, before we start that, we're going to start in a couple of minutes, okay? We're just giving some time for everyone else to join. I know some students are going to be 5 to 6 minutes late, okay? Until they start, I just want to give you a little bit of advice about what you should do, not just for tomorrow, but for the whole of your GCSE exam season, okay? Maybe some of you know this already, but some of you definitely need to hear this. Myself, I've been teaching for over a decade, okay? And I've taught hundreds of students. 3 advices that I would like to give you for this exam season is the following. Number 1, this season you need to up your game. So however many hours that you've been revising leading up to exam season, you need to double that. So if, for example, you've been revising 2 or 3 hours a day, in May, you need to double that, okay? Because if someone else, you got to think about this. You have to compare yourself with the other students that are in your year. Because you're literally competing against everyone else for the same GCSEs. Okay, everyone? So number 1, we need to step up the game. We need to make sure that the amount of effort you put in, whatever energy you've saved in the bank, that needs to be all out this exam season. That's the first thing. The second thing is that because your brain is going to be under so much stress, so much information is going to be coming into your brain, you need to rest well so that all that information becomes solidified and turned into memory. So what that means is, however many hours of revision you do during the day, try to rest well at night. Don't do all these all-nighters, okay? It's not going to serve you well. You have to rest well so that your brain can do its magic, and in the morning you wake up with all that information stored into your brain. Yeah. And the third thing is that you need to not give up. Okay. I've seen students change things around in the last couple of days. So right now we need to make sure that we don't give up. You can, if you're on a 6, you can turn it into a 9. Okay, guys. With that being said, should we start the live stream? Okay, fine. Well, look, guys, we're just gonna delay it for a couple of more minutes, another 10 more minutes. Tell all your friends who haven't joined yet. Okay, we're gonna delay for just 10 more minutes. Okay, a couple more people are joining. They haven't joined yet. What topics do you guys think are coming up? In the paper tomorrow? What do you guys think is coming up? Okay, we're gonna start. We're gonna start very soon. What else? If we haven't come to your school yet for TikTok, remind us. Put your school in the chat so that we can make sure that we come to your school. Okay, we've been to most schools. The most popular school so far was Southgate. If you're from Southgate, put W in the chat. If you're from Southgate got the most views at the moment. Almost 700,000 views. The second most. Oh, look at that! Look at all our Southgate students. There you go, W from Southgate. The second most popular school, from memory, was Hendon High. If you're Hendon High, put G in the chat for all the G's from Hendon High. If you're from Hendon High, put G in the chat. You guys got the second most views. Okay, look, we've got a bunch of Hendon High students here. The third most popular school from what I remember. Sufyan, what was the third most popular school on TikTok?
I don't know, but we went to Mayfields as well.
Oh, Mayfields. If you're from Mayfields, put M in the chat if you're from Mayfields. Mayfields was second most popular. Oh, sorry, third most popular. Mayfields. There we go. All the M's from Mayfields coming through. Okay, I think Cranford did quite well. They're about on 250k. All the Cranford students. If you Beale is literally coming out right now. Beale, if you're from Beale, your video was edited today, so that's coming out. We're gonna start in 5 minutes, guys. Okay, get ready. It's all gonna. If you haven't prayed, if you need to go pray Maghrib, go pray right now. In fact, I'm gonna go pray as well. I'll be back in 5 minutes. Okay, go. Wait, it's not even. No, it's not even time for Maghrib yet. My bad. It's not even time for Maghrib. Okay, fine. So, listen, guys, go eat something. Go use the toilet if you need to, because this is going to be an intense session. Okay, it's going to be absolutely intense. In terms of my advice for this session, there's no time for you to make notes, guys. Okay, we're not going to be making notes. Because why? I'm going to be going very quickly. So what I need you to do is this. I need you to just try to absorb the information, and if you got any questions, put it in the chat while we're going through the predicted topics. Okay, we're going to start in a couple of minutes, but if you got any questions, put it in the chat, and we're going to go through that during the live stream. But I'll be honest, you don't have that much time to make notes because we're going to be going very, very quickly, okay? And the good thing is that the whole thing is going to be recorded so that you can watch it again tomorrow morning just before your exam. All right, we're almost time to start. How many people we got on live right now, Zishan?
How many people got? TikTok live got 450.
And what about YouTube?
YouTube is 5k now.
5k, okay, 5k YouTube. Let's try. We're going to try and hit 7k live viewers before we start. Okay, everyone? Remember. Oh, one more thing. Okay, this is something that I was telling all my students earlier. That, you see, paper 1 is the most important paper in my opinion, because if you do well in paper 1, that means, let's say for example, paper 2 doesn't go that well. Yeah, let's say for example, paper 2, you know, a question, you fumble it, whatever, at least the average will still be high. But if you don't focus for paper 1, if you don't try your best for the paper 1s, and let's say paper 2 also goes bad, then you shot yourself in the foot. So in my opinion, paper 1s are always the most important papers, because from that you'll get the average, and it'll average out. Okay, cool. So get ready, guys. We're gonna start in literally 5 minutes. What are we on in terms of viewers for YouTube? 6k. Okay, fine. Tell all your friends to join, guys. Those who haven't got the link, tell them to join. Remember, it's live, but it will be recorded. Okay? You guys got this. Out of biology, chemistry, and physics, in my opinion, the most difficult one. Okay, physics is hard. Yes, everyone hates physics. But biology is going to be difficult in the sense that the questions are very, very peculiar. Okay? Cool. We're gonna start in 3 minutes, guys. Get ready. All right, cool. Fine. Should we just wait another 2 minutes? Okay, we're just waiting 2 minutes, everyone. Whoever needs to go pray it now. Come back and we're gonna start, guys. Okay, get ready. Get ready. Put your phones away. If there's a football match going on, turn it off, because this is going to be the most important lesson, the most important biology lesson of your life. Yeah. Also, if you have any topics that you want me to do after the live stream finishes, message, put it in the chat. We're going to go through it together. Okay. Again, if your friends haven't got the link, send it to them, because you're going to save their life. Don't gatekeep this. This is one thing you don't want to gatekeep. Okay, here we go, guys. All right. Is it my group? Okay, cool. Okay, I'll be back in 3 minutes. Okay, we're back in 3 minutes. Get ready, get ready. We'll be back in 3 minutes.
Boys and girls, join the YouTube live stream right now. The TikTok is streaming, but to see the content on the actual iPad, you need to go on youtube.com/ExamQA. Boys and girls, Mohammed is going to be a couple minutes. He's going to be a couple minutes. Share the video, share the live, get to as many people as possible. Make sure you like the video for the algorithm, so that we can begin and as many people can watch this as possible. I think Mohammed mentioned this earlier, but it's going to be very intense live stream. So to be honest, trying to write down everything he says is not the best idea. Rather, you should make very brief notes you need to be making, okay? If any at all. This is going to be posted as a recording after as well. That's when you can take the more sort of intense notes, more sort of fleshed out notes. But in this live stream, it's going to be very short, 1.5 hours, 2 hours max. So it's more for understanding, okay? We're going to be doing these live streams for paper 2 for bio, paper 1 and 2 for physics and chemistry as well, and for maths paper 1, 2 and 3. So make sure you subscribe. So that you can see these night before live streams for all the core papers for maths and science. Okay, guys, subscribe so that you can be notified of all these live streams we're going to be doing. It's completely free. All these live streams are completely free. So subscribe and get the notification when we do them. Of course, we're going to announce them on our TikToks as well. So, don't worry about that. On the TikTok tonight, we're just actually live streaming on YouTube, but TikTok is where we make the announcements. Okay? So, give Mohammed a couple minutes and we will begin. Okay? Again, this is completely free of charge. So make sure you can share it to anyone that's doing year 11, even year 10 people that are doing mocks. They can begin as well. They can tune in and benefit from this live stream. Okay. The live stream is mainly going to be for AQA content, but EdXL and OCR can join. Just be mindful of what is and isn't in your spec. Okay, Mohammed is here now, so we can begin. Make sure you lock in. This is time to lock in now.
Okay, guys, here we go. That's it. Get ready. Turn off every single distraction that you got. If the chat is distracting you, turn the chat off. Don't view it. Okay, do not view the chat if it's distracting you. Okay, here we go. We're going to start with the first predicted topic coming up. Okay, and 1 second, my iPad's loading. Get ready, guys. Okay, lock in. It's time to lock in. Maximum lock in. Okay, here we go. Turn off the chat if it's distracting you. Here we go. All right, so first chapter is going to be about the practical, where we investigate the function of an enzyme. Okay, so look, we know that the rate of an enzyme, how quickly an enzyme works, can be affected by either temperature or pH. Yeah, so we're going to start by looking at how the temperature of a reaction can affect the speed of an enzyme. So, first of all, the reaction that we're going to use is the reaction where starch turns into maltose. So starch is the reactant, maltose is the product, and the enzyme that we're going to use is called amylase. Yeah? So, for this reaction, we're going to also include iodine solution. And the reason behind that is because with iodine solution, there's going to be a color change, where starch is blue-black, you need to know that color, and maltose is browny-orange. Okay, guys? So the reason we're using a color change is because it will help us to monitor the speed of the reaction. Cool. So and of course we're going to do this reaction at different temperatures. Yeah. So we're going to do the reaction where starch turns into maltose. We're going to do it at different temperatures, and then we're going to see how does temperature affect the time it takes for the color to change. Okay, cool. So let's get straight into the practical. So step 1. Here's a practical method. Again, there's going to be a summary at the end of it, so you don't need to copy this down because I'm going quite quickly, okay? First of all, we're going to put some starch and amylase into separate boiling tubes. That's step 1. Step 2 is we're going to put those boiling tubes into a water bath, okay? So put them into a water bath. fill up with water, and then we're going to set the temperature to our first temperature. Let's say, for example, 20 degrees Celsius. And we're going to leave that on for 5 minutes so that both of them get to the same temperature. Then we take it out and we're going to mix the amylase with the starch. We're going to mix them together. And then we're going to add a couple of drops of iodine. And what's going to happen is going to turn the mixture bluey black. Okay? So this is our mixture. Now, then we're going to start a timer and start to transfer some of the solution onto a spotting towel and do that for every 10 seconds. Okay, so we're going to grab some starch and amylase, transfer it to the spotting towel, and we're going to repeat that every 10 seconds. Okay, and so when do we stop? We will stop when the color goes browny orange. So for example, for this reaction, it took 60 seconds for it to go from blue black to brown orange, which means that it took 60 seconds for the amylase to break down all of the starch into maltose. Yeah, so then you're going to record that. So for 20 degrees Celsius, it took 60 seconds for the reaction to stop. Then we're going to repeat the same practical with all the different temperatures. So for example, the first one was 20 degrees, it took 60 seconds. Repeat the whole practical with a new temperature. Let's say 30 degrees. Let's say that took 40 seconds. Then we're going to do 40 degrees. That took 30 seconds. And finally 50 degrees, there was no color change. So you might be asked to explain, why this color change is there. Okay, so first of all, so 40 degrees we said is the optimum temperature. Why? Because it took the least amount of time for the color to change, which means the reaction was fastest at 40 degrees. 20 degrees took the most amount of time for the color to change. What does that mean? It means it was very slow because the enzyme had low kinetic energy. You want to use that word, kinetic energy. Yeah? And finally, for 50 degrees, the color did not change. Why? Because the temperature was too high, which means that the enzyme was denatured, so it did not complete the reaction. Now, what kind of things should we control to make sure that this experiment is a fair test? The following. Number one, you want to control the volume of amylase and starch in every experiment. The second one is the concentration of amylase and starch. And the last one is the pH of the solution. And the way we do that is by using a buffer solution. Okay, so these ones are the control variables. Okay, guys? Now, here's a summary of that whole practical. In case I was going too fast, here's a quick summary. If they ask you, how do you investigate the effect of temperature? Here we go. First one, put starch and amylase into a boiling tube. Okay? I want you guys to screenshot this afterwards. Mix the starch, the amylase, and the iodine. Step 3, put the pipette the mixture onto a spotting towel and do this every 10 seconds. Step 4, record the time it takes from the color to go from blue-black to brown. Repeat and get an average. Look, in every practical, this is always 1 mark. Very easy to say. Repeat and get an average, okay? Finally, repeat with different temperatures and record the time. And finally, mention the things that you have to keep constant. So, volume of starch and amylase, concentration of starch and amylase, and the pH. These are the things that you need to keep constant. Okay, everyone? Cool. Moving on. We know that temperature is one factor that can affect the rate of reaction, but sometimes they might ask you about pH. Okay, so if they ask you about pH, it's the same summary. However, just add these things to it. Okay, so again, I want you to screenshot this. I'm not going to read the whole thing out, but if they ask you about pH, how does the changing the pH affect the rate of a reaction? This is going to be your summary. Okay, everyone? Again, when you watch it later, you can pause and write all of this down. But that's the first practical. Okay, moving on to the next topic. The next topic. Get ready, everyone. Is the chat slow now? Yeah. Okay, cool. All right, guys, get ready. How are you finding it so far? I hope you guys learned something. But time for the next practical. Osmosis. So here's the osmosis practical. First of all, remember that this is the definition of osmosis, okay? It's the oops, sorry guys. Osmosis is the movement of water across a semi-permeable membrane. So you want to mention that word, semi-permeable. So as you can see over here, we've got water on both sides, okay? And we've got sugar on both sides. However, there is more water on the left side. So because there's more water, we say that is dilute. Any solution that has more water is a dilute solution, okay? And of course, the other one you can say is concentrated. So water likes to go from an area where there is more water to an area where there is less water. Yeah? So there's more water on the left, there's less water on the right, so water is going to go from the left to the right. Yeah? And by the way, always remember the cue. Osmosis is passive. What does that mean? It does not require any energy. Okay. Moving on. So that's the first bit. Just remember, what is osmosis? It's the movement of water from an area where there is more water to an area where there is less water. Okay, so the practical that you could be asked about osmosis is the following. Find the water potential, or find the concentration of water inside a potato cylinder. Okay, so here's what we're going to do. Again, I'm going to go through the practical first, and then I'm going to give you the summary to write down. Yeah. What is it? Oh, it ended. Guys, can you hear me? If you can hear me, put W in the chat. If you can hear me, put W in the chat. Oh, they can still hear me. If you can see my screen, put L in the chat. Put L in the chat if you can see my screen. W. Yeah? Okay, fine. Alright, it's good, yeah? Just, what's it called? Have you got, like, you're viewing it live, right? Okay, calm. Alright. Okay, guys, I don't know. Apparently people were saying it ended, but it's good that we're on it, back on it. Okay, anyway. So the practical was, we're going to find the water potential of potato inside a potato cylinder. So step one is we're going to cut out some potato cylinders using a cork borer. Okay? Apparently it's lagging. Lagging hard? Okay. Okay, we're gonna keep going. Hopefully okay, if it's lagging, let me know. When it gets better. 1 second, apparently it's lagging. We're good, yeah? If we're good, just put a thumbs up so I know that we're good. Apparently it's lagging. Okay, fine. We're good. We're gonna continue. Alright, fine. Anyway, continuing. So use a mass balance. Okay, if you use a mass balance. So first of all, cut out the potato cylinders using a cork borer. Step 2, use a mass balance to measure the mass of all the cylinders. Okay, now don't worry if the potato cylinders don't have the same starting mass. Okay, don't worry about that, because we're going to use percentage change later. Yeah, so don't worry about if the potatoes that you cut out don't have the same starting mass. Then moving on, we're going to prepare sucrose solutions of varying concentrations. So there's going to be sucrose solutions which have different concentrations. Now, those potatoes that we cut out earlier, each of those potatoes are going to go into different beakers with sucrose. Okay, guys? Put them in there, and afterwards, we're going to leave them for a long time. Make sure that it's fully submerged. Yeah? Make sure that it's fully submerged so that the potato changes mass. Okay, now we can see that, for example, some potatoes will get larger. Yeah, some will get, oops, I spelled that wrong. They'll get larger. Some will get smaller. Okay, but we'll explain that later. Oh man, it keeps ending. Apparently everyone's saying it's ending. Apparently saying it's ending. Okay, guys. It's good. Something is good, something's ending, yeah? Cool, moving on. So then remove the potato cylinders, pat them dry, okay, to remove the surface water. So we're gonna pat the potato cylinders dry to remove the surface water. Yeah? And then after that, we're going to reweigh all the cylinders. Okay, guys? And finally, we're going to calculate percentage change in mass. You want to do it on my laptop? Everyone's gone, yeah? Okay, cool. Alright, moving on. Then, don't forget to include one experiment which doesn't have any sucrose, it's just got distilled water. Okay, that's going to be our controlled experiment. And finally, we're going to work out percentage change, okay? So, to calculate percentage change, let's say, for example, the potato cylinder had 5 grams before and 8 grams after, okay? We're going to do 8 over, take away 5, divided by 5. That will give you the percentage change. That will be plus 60, okay? And then after that, we're going to plot all of those different percentage changes onto our graph. Yeah. Now, for those of you who just want to know the summary, here we go. Here's a summary of the whole practical. So up to that point was just for you to understand the practical. Here's the actual summary that you need to memorize. Here we go. So I'm going to leave it on here for a minute so that you can all see it. Yeah. So we're going to leave it for a minute so you can all see it. While that is viewing, what's happening with the live? Is it good? Okay, guys. We can swap laptops if you want. You put on this laptop. It's okay. Cool. All right, guys. Fine. So I gave you some time. So that was that. Hopefully you all wrote it down. We're going to now go back to the next topic. Okay, here we go. What is it? Get ready, guys. Get ready for the next topic. Okay, the next topic, microscopes. Okay, so first of all, this is a light microscope. Yeah. Now on this microscope, you have to know the different parts of the microscope. Starting with, you have the eyepiece lens over here. Okay, so the eyepiece lens is where you put your eye. That itself has a magnification, sometimes of ×10. Then you've got the objective lenses, and these are different powers. So you have the ×10, ×100, ×1000. Okay, and then after that you have the light source. Okay, so the light goes through here. Then you've got the slide and the stage. Yeah, these are the different parts of the microscope. And finally, the two important knobs. Okay, so you've got the coarse knob, which moves the stage up and down. That's called the coarse knob. And then you've got the fine knob. The fine knob makes the image clearer. So for example, right now it's a bit blurry. So you change the fine knob to make it more clear. Okay, everyone? Moving on. Sometimes they might say to you, oh, cells will look transparent under a microscope. So what do we use to make the cells more visible? We're going to use a stain that will make it more visible. Okay, so the stain will allow you to see the cells better. Moving on. How do you prepare a slide for the microscope? Step 1 is place a drop of water on the slide. Then use tweezers to add the specimen. Okay. Finally, put a cover slip. And then put your dye. That's all it is. Okay, so I'll repeat that quickly. You're going to place a drop of water, add the specimen using tweezers, place a cover slip, and make sure there are no air bubbles. Okay, and finally you're gonna use a dye. With the dye, you can just say add a dye. You don't have to show it in this way, but just say add a dye. Moving on. Important calculation for microscopes. Magnification equals image size divided by actual size. Guys, if it's lagging and stuff like that, just try your best to focus. We've got a team working behind the scenes to fix it, okay? But just make sure you try your best to focus. Okay. I was just told that if it's lagging too much, try to lower the quality of the video so that way it doesn't lag for you, okay? Moving on. So back to this, yeah? So we have a magnification. So the formula for magnification is image size over actual size, yeah? Now, what does that actually mean? So the image size is what's going to be on your piece of paper, the thing that you're going to use your ruler to measure. That's image size, yeah? The actual size is what's the actual size of the specimen, okay? So for example, let's say you have a question like this. This cell has an actual diameter of 20 micrometers. Calculate the magnification. So how would you answer a question like that? Step one, remember magnification is image over actual, okay? Step two, you're going to get your ruler, okay? Imagine this cell was on your piece of paper. You're going to get your ruler, you're going to put it on the cell, and you're going to measure the length of the cell with the ruler. That length that you measure is called the image size, yeah? So that goes over there. The actual size, that's already given to you in the question. Remember, so another word they could use for actual size is real size, okay? So if they say real, it just means actual. So that goes at the bottom of the fraction. The final bit is to convert them so that both of them have the same units. Now, if you don't know your units off by heart, here's a table that you need to memorize, okay? So what I would say is just memorize one direction, and then you can, for the other one, is just divide, yeah? Continuing. So what we're going to do is we're going to convert this 5 centimeters into micrometers, yeah? So we're going to go from centimeter to micrometer, which means we're going to times it by 10,000. There you go. So that's 50,000 micrometers, and then you just divide them together, and that gives you a magnification of 2,500. Okay? So that is how to do a magnification question. Sometimes they might not give you the actual size. Can you see in the previous question, it says the actual size in the question, but sometimes they might not give you that. Instead, they'll give you something called a scale bar. So if they give you a scale bar, here's what you're going to do. You're going to, number one, again, magnification is image over actual. Okay, so grab a ruler and put it on the scale bar. The length of the scale bar in your ruler, that is your image. Okay, so whatever your ruler says, when you put it against the scale bar, that is your image. The number on the scale bar, that is the actual number. Okay, so that's the actual size. Does it make sense, guys? So whatever the ruler says, that's your image. Whatever number's on the scale bar, let's say it says 15 micrometers, that is your actual. So once you've got that, don't forget to convert them to make sure. And so many people make this mistake. Make sure that the units are the same. It doesn't have to be centimeters, but as long as they're the same, then you can divide them together, okay? And there you go, you get your magnification. So that's that part. Quickly moving on, I'll skip this part. So I'll skip this as well. Here we go. There are two main types of microscopes, okay? You've got the light microscope and the electron microscope. They could ask you to compare the differences between them. So I'll just tell you the one about the light microscope. Here we go. Number one, it produces a color image. Number two, you can view living specimens. Whereas the electron microscope, the specimens will die. Yeah? Next one. It does not require a lengthy preparation. You can prepare it very quickly compared to the electron microscope. It's cheaper. Okay, so these are all the advantages. Moving on to disadvantages, okay? Here we go. Lower resolution, lower magnification compared to electron microscope. And finally, you can't see small organelles. The only thing you can see with a light microscope is the nucleus. That's the only thing you can see. All the other smaller stuff, you cannot see with the light microscope. Okay? So here we go, guys. Moving on to the next topic. We're doing very well. What time is it? It's 9:05. We're going to try and go for 1 hour. Okay? After 1 hour, that's it. Finish. You guys can go to bed. Here we go. Next chapter. We're going to go through osmosis now. Sorry, my bad. Mitosis. Okay? Chat, how are you guys finding biology? Are you ready for bio tomorrow? You guys ready for it? What I'll do is, first, I'm going to go through all the predicted topics, okay? After that, I'm going to take requests. So if you guys have any topics you want me to teach you, we'll do that afterwards, okay? But first, let's go back to the next predicted topic, which is mitosis. Here we go. Cool. So what is mitosis? When a cell divides to make 2 identical copies. Okay, everyone? So don't confuse that with meiosis, where you make 4 cells in mitosis, you make 2 identical copies. What is mitosis used for? It's used for growth, repair, and replacement. Okay? So in mitosis, a parent cell makes 2 identical daughter cells. Okay? Now, before we get to the summary, let's go through the process of how it works. Okay? Step 1. You have a cell cycle, like the life cycle of a cell. The longest part is called interphase. Yeah? During interphase, the main things that happen is the DNA and the organelles duplicate. Okay? So the DNA, everything duplicates. Number 2, we have mitosis. What happens during mitosis? Let's have a look. So number 1, obviously in mitosis we know the whole division happens, but how does it happen? Here we go. Step 1, DNA coils up to form X-shaped chromosomes. That's the first step. Yeah, there we go. We have the X-shaped chromosomes. They're held together at the middle. Okay, the arms are called sister chromatids. These are the arms, and they're identical. Moving on. Step 2, the chromosomes line up in the middle of the cell. They line up in the middle of the cell. Step 3, centrioles, which are these little guys. Think of them like little Spider-Mans. They're going to shoot fibers called spindle at the chromosomes. And then they're going to pull back. The chromosome arm. So step 4, spindle pulls back the sister chromatids like that. Once they've been pulled apart, a nucleus is going to form around the chromosomes, okay? And then it's going to divide, just like that. There we go. So, summary for mitosis is right here. I'll give you guys 30 seconds. Just make sure that if it comes up, you use the exact words that are on the screen, okay? Don't use different words. Try to use it word for word, okay, everyone? So that's mitosis. Moving on to the next topic. We're going to go through stem cells. Get ready. We're trying to finish on time so that you guys have a good rest at night. Okay. Yeah, here we go, guys. Get ready. Next topic coming up. Quick one. Stem cells. Here we go. So, if they ask you what are stem cells, what's the definition of stem cells? It is the following. Stem cells are unspecialized cells which can differentiate and turn into all different kinds of cells. Yeah, that's basically what stem cells are. Yeah. Now, in animals, you can get them from 2 places. One is the embryo. Anyone else know where else we can get stem cells? One is the embryo. Where else can you get them from? Let's see who's the first person to answer it. First person to answer it. Here we go. I'll shout them out. Bone marrow. Well done, Verm. Okay, Verm got it right. So you got the embryo and the bone marrow. Okay, so that's where you get stem cells in an animal. Okay. Now the embryo stem cells can turn into any type of cell. But guys, ExamQA mod is getting okay. Put W for ExamQA mod. Anyway, back to this, yeah? So, embryonic stem cells can turn into any type of cell, yeah? But the stem cells from a bone marrow can only turn into blood cells, so like red blood cells and white blood cells, okay? So which one do you think is more useful? Is it the embryo or the bone marrow? The embryo, yeah? Because they are so useful, doctors like to use them for medicine, okay? Why? Number 1, they can be used to replace faulty cells and turn them into healthy cells. That's one benefit of embryonic stem cells. Number 2, okay, so for example, I'll give an example of that. Let's say someone's got diabetes, we can use stem cells to make them new insulin-producing cells, okay? Secondly, it can treat things like paralysis. Let's say someone's got damaged nerves, we can use stem cells to create new nerve cells for them, so that way we treat them. Okay, everyone? Now, stem cells are not rejected by the host. So, for example, if you use your own stem cells, if the doctor uses your own stem cells, then your host cells are not going to try to reject them and kill them. Okay, everyone? That's another benefit. Cool, moving on. And this process, where we use stem cells to treat diseases, is called therapeutic cloning. Okay, everyone? That's the definition of therapeutic cloning, where you use your own stem cells to treat diseases. Who's that? Oh, TikTok. Oh, okay. Hello, TikTok. Come on, guys. Jump on YouTube so I can see your comments. Moving on. Now, it's not all sunshine and rainbows because we've got some disadvantages as well. Yeah? What's the main disadvantage? Ethical issues. An embryo has two pathways in life. It can either become a baby and become a full human being, or it can go in the bin, right? So people say, oh, we're destroying potential life. Okay. Secondly, is that those stem cells could get infected by viruses during the whole process. So those are the two disadvantages. Number one, we said ethical issues, and number two, we said that it could get contaminated. All right, moving on. So some people say, no, it's fine, use stem cells, it's okay. What do they say? What's their argument? The first argument is it cures sick people, okay? And the second one is that we're using embryos that the parents didn't want in the first place. So it's okay, they were going to go in the bin anyway. We're using them to treat people. Yeah, so that's the argument for using stem cells. Make sure you remember these two arguments, guys. Next one. The arguments against stem cells are, number one, we're killing potential life. Okay, and number two, you could use alternative treatments. That's the arguments against it. So whether it doesn't matter what side of the fence you guys are on regarding this topic, whether you're for or against it, just make sure you remember the two arguments for and against stem cells. Okay, guys? Now, plants also have stem cells. They are found in the meristems, which are found in the tips of the shoots and the tips of the roots. So if they ask you, where do you find animal stem cells? In the bone marrow, in the embryo, okay? Where do you find plant stem cells? In the tips of the roots and the shoots, called the meristems, okay? Now, the one final difference is that plant stem cells are there for its entire life. 24/7, it will always have it. So that means we can use it to grow more plants and prevent extinction. Good. Stem cells done. Moving on to the next topic, guys. Moving on to the next topic. Okay, so a very quick one. A very quick and easy one. Make sure you get my good side on TikTok. Alright, guys, come on. We got this. Remember, biology paper 1 is the most important because it's going to set the tone for the rest of your exams. Here we go. Food tests. Food tests are basically where we go through the different ways you can test for different types of food. Starting with protein, okay? So if you got a test tube and you think it might contain protein, the way you confirm, okay? What school was the protein sort of film? Do you remember? Oakwood, guys. If you remember this one, when I came to do TikTok with you, protein. What was it? You add burette. Okay. Now, if you add burette for a protein, it goes purple if there is protein. Okay. And if it doesn't go, if there's no protein, then it just stays blue. Yeah. So that's burette. B for burette, B for blue, P for protein, P for purple. That's how you remember the protein one. Good. Moving on. Moving on to starch, yeah? Sometimes the moderator makes typos in the chat, okay? Ignore what he said. Alright. So, for starch, we're going to add iodine, okay? We're going to add iodine. So, iodine, if there is starch, it goes bluey black, as you guys remember from the enzyme practical. And if there's no starch, it will go brown. There you go. Yeah, that's it. Starch, easy one. You add iodine, it goes blue-black if there's starch. If there's no starch, it goes brown. Next one. For lipids, if you guys do AQA, for lipids, it's the following. Sudan 3. If there's lipid, okay, you're going to form 2 layers. The top layer goes red. If there's no lipid, the whole thing goes red. Okay, guys, that's for lipid. If you're not AQA, you can do the emulsion test. For example, Edexcel. What you do, you add ethanol, okay? You mix it up. You add a bit of water. And if there is lipid, it forms an emulsion. So basically, it will go cloudy, yeah? And if there's no lipid, it will just go blue. And the last one, Benedict's is for sugar, yeah? For glucose, simple sugars. We're going to use Benedict's. So Benedict's, if you add it and there's sugar, it goes the following colors: green, yellow, red. Yeah, those are the colors depending on how much sugar there is. If there's a lot of sugar, it goes red. Just remember traffic light, like a traffic light. And then if there's no sugar, it goes blue. That's it. All right, everyone? Finished. Next topic coming up. Remember that these are things that you guys should know, but hopefully it's a revision for all of you. We're going to go through the next. This is a big one, okay? The circulatory system. AQA don't do ethanol. Ethanol was only for EdXL and I believe OCR. Yeah? AQA do Sudan 3 for lipid test, in case you guys are wondering. Moving on to the circulatory system. Okay, first of all, our heart pumps two circuits. The first circuit takes deoxygenated blood, so it means it's got blood that's full of carbon dioxide, to the lungs, yeah, and it brings back oxygen to the heart. Then it will pump again to the body, okay, carrying oxygen, where the cells of the body will use it for respiration and bring back the deoxygenated blood. So if they ask you what is a double circulatory system, just say it's two circuits that are joined together, and that's what we have. Moving on, we have to know the structure of the heart. Okay, if you like this picture of the heart, put W in the chat. If you like this drawing, yeah. So, structure of the heart. Following. This is you're more likely to get this one in the exam. Okay, so we're gonna go through that quickly. First of all, the heart is flipped. Everyone, it's flipped. So the right side is on the left side of the page. Okay, and the left side of the heart is on the right side of the page. So make sure you don't confuse that, guys. Yeah? Cool. Anyway, moving on. Veins bring blood into the heart. The veins will bring blood in. So on the left side, we have the vena cava, which is bringing blood from the body. Okay? And I colored it blue so that you guys remember, blue means it has carbon dioxide blood. Yeah? Deoxygenated blood. It does not contain oxygen. On the right side, we have the pulmonary vein bringing blood in, okay? So on the right side, it's blood that contains oxygen, yeah? So blue means deoxygenated and red means oxygenated. Moving on. So after that, we have the two chambers near the top. They are called the atria, okay? We have the right and the left atrium. Then we have valves to prevent backflow. So if they ask you what is a valve, say it prevents backflow, yeah? So you have the atrioventricular valve, very big word. Then we have the bigger chambers, the ventricles at the bottom. Okay? Finally, so the blood goes like this. It goes down to the ventricles and then it's going to shoot up out of the heart. So it's going to come down like this and it's going to shoot up out of the heart. Okay? So next is the semilunar valves. And finally, we have the arteries that take blood away. Yeah? So we've got the pulmonary artery, which is taking blood to the lungs. That's the pulmonary artery. And we've got the aorta, which is taking blood to the rest of the body. Done. There you go. Structure of the heart complete. One final thing. You see over here, the left side of the heart is slightly thicker. Can anyone explain why it's slightly thicker? Did I draw that by mistake, or is that a real reason? Why is it slightly thicker on the left side? Go on, first one to answer it. Go. Raw. Why is it thicker? Come on, let's have a look. Oh, that was fast. I can't even see who said it first. But well done. It's thicker for two reasons. To pump blood with more pressure so that the blood can go further away. Yeah? W. Alright, moving on. Next part, the pacemaker. Okay, so the pacemaker is found. First of all, what does it do? The pacemaker, also known as the SAN, controls the heartbeat. Okay, that's what the pacemaker does. Where is it found? You have to know where it's found. It's found in the right atrium. Now, sometimes your pacemaker can get damaged. If it gets damaged, do we give up? Is that it? Game over for us? Life over? No, because we can use an artificial pacemaker to help control the heartbeat. However, here's the crazy part. If you have an artificial pacemaker in your heart, okay, you have to avoid magnets. You have to avoid them. Why? Because it could damage it. Yeah? So that's a very key thing. If they ask you, Bob had an artificial pacemaker, why does he have to avoid a magnet, such as an MRI? Yeah? Because the MRI magnet could rip it out of his heart. Yeah? That's it. Good. Moving on. We have three types of blood vessels. the following: artery, vein, and capillary. Yeah? So arteries and veins are the big ones, and capillary is the small one. Let's quickly compare them. You could get a 4-marker comparing the structure of the artery versus the vein. So first of all, let's go with the layers. The first layer is called the endothelium, which is 1 cell thick. Okay, you have the hole in the middle, which is called the lumen. Yeah? Then you've got a smooth muscle layer around it, which stops the vessel from bursting. Yeah? Then you've got the final outer layer, which is collagen, which makes it tough and strong. So arteries and veins have these 4 layers. So what was it? Lumen, endothelium, smooth muscle layer, and then collagen. Yeah? Let's continue. Now, let's compare the structure of artery versus vein. Okay, chat is getting wild. This is some mad stuff. Okay, moving on. So, artery versus vein. So we have, the first difference is that the pressure in the artery is much more higher. Why? Because of heart contraction. That's the first one, okay? Number 2, the artery has a thicker layer of muscle, again, to withstand the higher pressure, okay? Number 3, the artery has a smaller hole in the middle. What's the hole called again? Lumen. It has a smaller lumen, yeah? So the 3 differences was, 1, it was high pressure in the artery because of heart contraction. Number 2, thicker muscle layer. And number 3, smaller hole, smaller lumen. Yeah? Now, the difference is that the veins also have valves. So that's the 4th difference. Okay? Veins have valves to prevent backflow because the what? The blood pressure is much lower. Okay. Moving on. So let's continue with the next one. Capillaries. Capillaries are the smallest blood vessel, and their main job is to exchange materials. That's it. That's capillaries. Okay? Done. Moving on to the next topic. Okay, while that loads up, chat is apparently going wild. Is that true? Nishan, they going wild? They're finished. Chat is finished, apparently. You guys need to pattern up. What is going on? Worse than last year? Oh my god. Can't beat last year. Last year was crazy. Okay, let's have a look. Enzymes, done that. Next one coming up. Get ready. Alright. So, we're going through a board. I'm going to screenshot this. Whoever puts the worst comment, screenshot, name and shame on TikTok. That's it. Wait, is that going to motivate them to make... Oh, okay. No, don't put bad comments. Alright, here we go, guys. Get ready for the next one. Yeah, this is a boring one. It's about drugs. Yeah, no one likes drugs. Here we go. Let's go. First one. What is a drug? It's a chemical that changes how the body works. That is a drug. So those of you who are asking how much longer, maximum 20 minutes longer, and then after that Q&A, and then after that triple science, and that's it. Finish. Yeah, cool. So the 3 types of drugs are painkillers, which reduce painful symptoms. Okay, you've got antibiotics, which kill bacteria, not viruses. And finally, so just remember, antibiotics, B for biotics, B for bacteria. And finally, antivirals, which kill viruses. Yeah, those are the 3 types of drugs. Remember that antivirals do not kill bacteria because viruses hide inside your cells. Okay, now a very quick look at antibiotic resistance, which is not good for us, but very good for the bacteria. Step one. Let's say a person becomes infected with bacteria. You take some drug. So he goes to the doctor, goes, I'm infected. Take some antibiotics, okay? That helps him. So now he's slightly feeling better because some of the bacteria got killed. Because of that, he stops taking the antibiotics. Is that a good thing to do? Definitely not, because the remaining bacteria are going to replicate. Okay, so those bacteria are going to replicate, and now he's ill again. So what does he do? He runs to the kitchen, tries to find the antibiotics again, the same antibiotic as he used initially, but it's not going to work because all the bacteria have become resistant. Okay, guys? So what does he have to do now? What can we do? 2 options. So if they ask you, how do you kill resistant bacteria? Number 1, use a stronger dose or a new antibiotic. Yeah? There you go. Now, let's say, how can we prevent antibiotic resistance? So those of you who want to be doctors, this is very important, okay? If you're a doctor and the patient comes to you and says, I'm ill, can I have some antibiotics? Here's what you do. Number one, tell them to finish the course if you do give them antibiotics. Why? Because if they don't finish the course, then what we just spoke about earlier will happen. The bacteria will become resistant. Number one, finish the course, okay? Number two, do not prescribe antibiotics for simple illnesses. So if someone's got a very mild illness, you don't have to pull out the big guns. You don't have to pull out the antibiotics. You can just say, look, have some rest and your body will kill it by itself, okay? So two ways to stop antibiotic resistance. All right, next part. Where do drugs come from? 3 places. So all these drugs that we have in the supermarkets and the pharmacies, all these drugs come from 3 places. Number one, from plants, okay? And there are 2 examples from plants that you need to memorize, guys. Here we go, word for word. First one, aspirin is a drug. It's a painkiller, and it comes from the willow tree. Next one, digitalis. It treats heart conditions, and that comes from a plant called the foxglove. There you go. Those are 2, no more than that, just those 2, okay? Some drugs come from microorganisms, okay? So, for example, penicillin is an antibiotic which comes from fungus and also from other antibiotics. And finally, we've got drugs that are made by the pharmacists. Big up pharmacists around the world, okay? Synthetic drugs, that's it. Done. Okay, moving on. That's it, moving on. Come on, guys, stay with me. Don't lose focus. You had the whole day to rest. Oh, we're not even on 2K likes yet. Guys, hit the like button. We need to hit 2K likes because we're low on likes at the moment. Let's continue. What is on? Next topic coming up. Next topic is, we're going to go through the immune system, guys, very quickly, okay? Don't give up on me. Stay focused. Go drink a coffee if you need to. Get ready. Next topic coming up. Okay, immune system. So, we've got, in our immune system, which is the system that our body uses to fight pathogens, there are 2 parts to it, okay? We have physical and chemical barriers, and then we've got the actual immune system itself, yeah? So, let's go through the physical and chemical barriers first. The first one is our skin, which is very tough for penetration. Bacteria can't go through our skin. Number 2, mucus in the airways to catch those bacteria from going any deeper. And finally, we have hairs that will remove all the mucus out of your body. Okay, if you don't have cilia, if your cilia don't work, the mucus will stay in your body. Okay, those are your physical barriers. Chemical, you've got stomach acid. There's acid in your stomach, so even if you, let's say sometimes you drop your food on the floor, you pick it up, 5-second rule, you eat it, it's okay, you can't do that because your stomach has acid inside it. Alright, moving on. Next part. In our teas, there are enzymes which helps to break down the pathogens. Yeah? Continuing. So that was our physical and chemical barriers. Then we have the actual immune system. Who knows what's the name of the main cell that the immune system has? What's the main cell of the immune system? How do I get rid of this little voting thing? Raw. Sorry, guys. What's the main cell of the immune system? Sorry, guys. It's called the white blood cell. Okay. Now, the white blood cell does 3 things, and I want you guys to memorize this. It does 3 things. The first one is it does phagocytosis, which means that it can. What is phagocytosis? It binds to the pathogen. So make sure you include the word bind, okay? So it binds to the pathogen. Because why? Because the pathogen has foreign antigens on its surface and it swallows it up. There we go. So very quickly, one more time. Phagocytosis is when the white blood cell binds to the pathogen, okay? And it swallows it up. Good. Thank you. Oh, can't even see the thing. Now what's this? Oh yeah, okay, come, got it. Moving on. Step 2, antibodies. For whoever said this is not AQA, I feel sorry for you, mate. Just memorize it now, okay? All you've been playing Fortnite, just memorize this. Antibodies. Antibodies, okay? So the white blood cell shoots antibodies to the bacteria and destroys it. Okay? There we go. That's the second one. Antibodies are produced by the white blood cell. And finally, antitoxins. So let's say you had guests over, and now they go home. You have to clean up after the guests, right? You got to run to the dinner table and eat all the chocolates that you couldn't eat because your mum was watching you. Now that's where the antitoxins come up. Yeah. So the antitoxins are going to remove all the toxins from the pathogen. So what was it? Number one, phagocytosis. Guys, stop commenting funny stuff because he's gonna laugh. I'm gonna laugh with him. Yeah. What was it? Phagocytosis. Number two, what was it? Antibodies. And number three, antitoxins. Good. All right, we're gonna move on. Quick one. Vaccinations. Here's a quick summary of vaccine. I'm not gonna read it out. Okay. I'm gonna leave it here. You can come back to it later. But this is a model answer for vaccinations. Yeah. Let's continue now to the next slide. Raw. You guys saw all my folders now. You see all the secrets of ExamQA. Okay, so done that. We're going to move on to the final two topics. That's it. The final two topics left. Starting with respiration. Okay? I know everyone's getting tired, but you guys are doing very well, okay? Just two topics left. Try to stick through it, and then you can just go straight to bed, yeah? I know none of you are going to go to bed, but go straight to bed after this, yeah? Then, we're going to do triple science. Get ready. Also, make sure to like the new video that we just dropped. It was Beale School. If you're from Beale, put W in the chat while this slide loads up. If you're from Beale. I heard Beale's better than Mayfield. Is that true?
Write what school you are in the chat.
Yeah, write whatever school you're from the chat, yeah? If you're from Beale, W in the chat. All right, here we go, guys. Next one, respiration. So, respiration is when energy is released from glucose. Okay, and that happens inside the mitochondria. So that's respiration. Right, so mitochondria is going to use glucose to release energy. Does that make sense? It doesn't happen in the ribosome, mitochondria. Now, the energy that is produced is used for three things by the body. First one is to join monomers. Big up Northolt for this slide. Remember, guys, Northolt? Not monomers to form polymers. Okay? The second one is the energy from respiration is allowed for your muscles to contract. And the third one is that energy is used by mammals to maintain your body temperature. Yeah? So what was it? For joining monomers to polymers. Number two, to keep you warm. And number three, to contract your muscles. Okay, now there are two types of respiration. I hope you guys remember this, please, okay? Number one, aerobic respiration, which means with oxygen, and the other one therefore must be anaerobic, without oxygen. So those are the two types of respiration. Now, anaerobic actually is slightly different in an animal and a plant. Yeah? So let's start first with aerobic, and then we'll go down this crazy path over here. So aerobic, very easy. Who can tell me very quickly? First one, what's the equation for aerobic respiration? Go, quick. First one to write it down. Go. Quick one. First one, aerobic respiration. What's the formula? What's the formula? It was Ion. Well done, Ion. See, those fingers are fast, Ion. Well done. Keep going on. So glucose plus oxygen. Okay. Whoever said yeast is finished. Glucose plus oxygen makes carbon dioxide and water. Okay, everyone? So that's the one you need to memorize for aerobic respiration. And a good way to memorize that is, look, you have 6, 6, 6. Yeah, and that over here is the formula for glucose. Okay? Glucose plus oxygen makes carbon dioxide and water. Now, that's aerobic. And of course, don't forget energy as well. Yeah? That's the whole reason we're doing it, to release energy. Okay, anaerobic, starting first with animals. Glucose turns into lactic acid. Okay, that's animal one. Now, why did I put the scary face over here? Because lactic acid is actually toxic. Those of you who play sports, or whoever runs around, yeah? When you run around and you get a cramp, it's because there's lactic acid in your body. Okay, that gives you muscle fatigue and pain. So that's in animals. But is it the same in plants and yeast? No, it's not. Okay, so anaerobic makes less energy than aerobic. Yeah? So why do we even do it? If lactic acid makes less energy, why do we do it? Because we only do it during emergency situations. Okay, everyone? Now, what about in plants? How's anaerobic in plants? Glucose becomes ethanol and carbon dioxide. There. That's the one in plants and yeast. Who knows what this is called? It begins with F. It's the famous F word in biology. Who knows what it is? Come on, what's the name of this reaction? Oh, L-eclipse. Well done. Your name's too long, but well done. It's called fermentation. Well done, guys. This reaction, if they ask you, what is fermentation? It's this one. Okay, in plants. So now, glucose and carbon dioxide are used by humans for different things. Let's start with let's start with ethanol. Ethanol is used to make drinks like beer. Okay, antiseptics. So the stuff that you have in those hand lotions, hand sanitizer, and medicines. Yeah. Carbon dioxide, on the other hand, is used to make bread rise. So if you ever get a loaf of toast and you slice it up, the loaf of toast, you look at it you see those little circles. That's because of the carbon dioxide. Yeah. All right. Next part. Final part of respiration. Changes in your body during exercise. Yeah? What's the changes? What happens to your body during exercise? During exercise, two things happen. Number one, your heart rate increases. And your lungs go deeper and faster. So your breathing gets deeper and faster. Nothing else. Well, that's the main stuff, yeah? So, why does this happen? One very simple reason. Because it helps to increase the amount of oxygen going to your cells and to remove the carbon dioxide from your cells and out of your body. There you go. So your heart beats faster. Your lungs go deeper and faster to get in more oxygen and to release more carbon dioxide. Just for one reason. So you can make more energy. Okay? Done. Now, next part. When you do too much anaerobic respiration, yeah, you have something called an oxygen debt. So that means you burnt the glucose, you used the glucose without oxygen. That's called an oxygen debt. So you owe yourself oxygen. So what you do is, how do you pay back this debt? Well, two things. After you finish exercising, you will continue to breathe a little bit more. Okay? So, for example, imagine you're running after the bus, you run for the bus, you jump onto the bus, okay? And now you're breathing deep and heavy. Why? Why are you breathing deep and heavy? Because you want to do the following. You want to get more oxygen to your cells so that you get rid of the lactic acid. Yeah? Now, this question, if it comes up and you guys get this right, you'll be amongst the top students in the whole country. 2 ways that lactic acid is removed from the body. I want you to pay attention, okay? 2 ways. Number 1, it's reacted with oxygen to form carbon dioxide and water. That's one way. Who can tell me the second way? Let's see whose school has done them justice. What's the second way? Very good. Arafat Khan. Come on, W. Arafat Khan. The second one is the lactic acid is transported to the liver and turned back to glucose. Good. Well done. Yeah? So make sure you remember this, everyone. That's it. Respiration done. Moving on to the last topic for science. And that's it. You guys can go after this. Okay? Last topic. Yo. Yeah. Calm. Okay. When I say lost topic, I mean not really lost topic, yeah? Alright, so photosynthesis. There's 2 parts to this, yeah? There's this part and there's a practical. That's it. After that, done. Okay, photosynthesis is when light energy is used to make useful substances. That's photosynthesis, yeah? Where does it happen? It happens inside the chloroplast. So, here's a reaction. What do you guys notice about this reaction? If you have noticed it properly, it's literally the reverse of aerobic respiration. So if you know aerobic respiration already, and you flip it the other way around, it's photosynthesis. But don't forget, it also needs chlorophyll. That's one difference. And obviously it needs light. Okay? Happens inside plants. Okay, Happens inside chloroplants of chlorophyll. So glucose is used by the plants in 5 different ways. Let's see if anyone from Whitmore remembers. Whitmore boys, do you remember? What was it? What was it? It was number 1. If you don't remember, guys, if you don't remember, here we go. First one is used for respiration. Yeah. So we're talking about the 5 uses of glucose. Number 1, for respiration. Number 2, it's convert. When I said, don't say make, by the way, say release. Yeah, that was a typo. I should have said release. Converted into cellulose to make the cell wall. Number 3, converted into lipids and stored in the seeds. Number 4, glucose is combined with nitrates to make proteins. And the last one is that some of the glucose is stored as starch in the roots. Okay, what a beautiful diagram on your screen. Screenshot it, but more importantly, memorize that, guys. 5 uses of glucose. Okay, smashed it. Let's continue. That was Whitmore. Okay, now they might ask you, why does the plant take glucose and store it in the form of starch? Why can't the plant just store glucose as glucose? Why does it have to turn it into starch? Here's why. Okay, because starch is insoluble. So if they ask you, why does it happen? Starch is insoluble, which means that even if it rains, okay, even if it rains, it does not affect the plant because the starch is not going to swell up. Okay, everyone? Perfect. Now, that's that part. We're going to quickly go over the practical of photosynthesis. And then, come on, guys, come on, guys, come on. You guys are doing well. Keep it up. Don't lose attention. Here we go. Next one. Measuring photosynthesis. So, how do you measure the speed of photosynthesis? How do you measure it? So, step 1, we're going to get a pondweed. Okay? We're going to take that pondweed and we're going to connect it to a capillary tube and a gas syringe. So, over here is your capillary tube and over here is the gas syringe. Yeah? So, why is gas involved here? Because we know that the plant, when it does photosynthesis, what gas does it release, guys? What gas does it release? Go on. What does it release? It releases oxygen, right? So we know that when the plant is releasing oxygen, that's going to go and it's going to push this little bubble over here. There's an air bubble, okay? That bubble is going to move. And the faster it moves and the more it moves, the higher the rate of photosynthesis. Yeah? Okay, so step 2 is we're going to place a white light source, white light, near the pondweed. Okay, now when we use the white light, we have to make sure it is an LED light. Why? Because it does not release any heat. Yeah, so you have to use an LED light, not a normal bulb, because you don't want to release any heat from it. Okay, and if you don't have an LED light, you can just put a piece of glass in front of it so that the light can go through, but then the heat is captured by the glass. Yeah, all right, moving on. Measure the distance moved by the air bubble. That's step 3, easy one. Okay. Step 4. The distance moved by the bubble. We know that the more the bubble moves, the higher the rate of photosynthesis. Okay. Now, what are some control variables in this investigation? What do we control? Obviously, we know our independent variable. What does independent mean again, everyone? The one that you change, which goes on the x-axis, is what? It's the distance of light. We're changing the distance of light, okay? And what is the dependent variable, the thing that we're measuring, which goes on our y-axis? It's the rate of photosynthesis. In other words, how much oxygen is produced. So we're looking at how much distance the bubble has moved, okay? So that's your independent and that's your dependent. But we also have control variables, which are temperature, amount of carbon dioxide. So you want to make sure that the amount of carbon dioxide is always the same, and you can do that by adding a chemical called sodium hydrogen carbonate. Yeah, the amount of time that we let the reaction happen for, and finally make sure that you use an LED light. Cool. Last bit actually, I think that's it. So we're gonna do a quick Q&A just to ask anyone if they have any questions. Okay, so guys, if you have any questions, now is your time to put it in the chat. Before we do that, Zishan wants to quickly say something to you guys, so I'm gonna hand it over to Zishan. and then after that, yeah, bang, we're gonna do our Q&A. Okay, here we go, guys. What do you want, Zishan?
Yeah, calm. Let me chat to him.
All right, quick. Zishan's gonna speak to you guys, and we'll come back and do
Put your
Q&A.
questions in the chat. Put your questions in
In
the
the
chat.
meantime, put all your questions in the chat.
Put your questions in the chat. We're gonna have a quick 2-minute break while you put questions in the chat, and we're just looking at them. You're doing just look. Basically, because it's going so fast, as soon as you scroll up, it pauses it, so you can. Oh, that's all right. Okay, boys and girls, that was very intense. That was very, very intense. Remember, we're doing these paper night before live streams for every single paper. Mohammed is coming back, so don't worry. We're doing these night before live streams for every single paper 1 and 2 for all sciences, paper 1, 2, and 3 for maths as well. Okay, so subscribe so you don't miss the live streams. Okay, subscribe, like the video as well, so that you know it's helpful for us, it's helpful for the algorithm. I'm gonna let you in on a secret as well. If you check the pinned comment, code livestream will give you £10 off our current 2024 exam season package. Okay, all you have to do is you go to ExamQA.com. Okay, you click the middle package, 2024 exam season package. As soon as you click that, you click I have read and agreed the website terms and condition. Coupon code live stream. Okay, live stream. This is an exclusive offer. You click apply, and then you get £10 off. This is only for you guys. We haven't announced this code anywhere else. Okay, so all this content, all the slides you saw Mohammed use, okay, you get that for only £29. Now you might be thinking, exams are tomorrow. What is the point of doing that? What is the point of signing up? For physics, bio, and chem, you still have paper 1 and 2. With our resources, you can literally learn. I'm not even going to lie to you. Mohammed will back me up on this. You can learn the entire specification in literally a couple days. You've seen our TikTok content where we managed to teach these guys, you know, these finished guys, yeah. Apologies for them lot. They are very nice people, yeah, but they're finished. We managed to teach them in literally 1 minute. Okay, imagine you had these slides, these resources, these notes to yourself, right, for a whole month. There's still a month left of exams. Think about the amount of content you can learn and guarantee yourself in terms of the grade. What grade can you guarantee yourself if you had this subscription? Okay, so that's an exclusive code for you guys, code livestream. Use that exclusive offer only. And in fact, at the end of tonight, I'm actually going to sort of cancel that code, okay? So it's just an exclusive one for you guys. Mohammed, back to you. Thank you very much for your time. And good luck for the rest.
Okay, everyone. So, got a quick pump because it's about to get serious here. All right, so, one second. So what did you guys say? Inverse square law, okay. Fine, we'll do the inverse square law, and after that we'll do a couple more topics, okay? And after that we're gonna go to triple science. So triple science, guys, get ready, your time is coming up. Okay, here we go. Inverse square law, yeah? So basically, the light intensity that the plant experiences is proportional to 1 over d squared, okay? Now I'll show you exactly what that means and how to use it in the exam. But this is what you need to understand. That's the formula, okay? So for example, what does D stand for? D stands for final distance divided by initial distance. That's the formula, yeah? So let's apply it here. Let's say, for example, the LED light was 20 centimeters away, and then we moved it closer. So from 20, we brought it closer to 5 centimeters. Okay, guys? Now, how much would you say that the light intensity has increased by? Well, here we go. We're going to do final distance. What was the final distance? What do we say? It was from 20, it went to 5. So the final distance was 5, okay? And the initial distance was 20. So 5 over 20, which is what? 0.25. Okay, that's the value for D. And then you're just going to do 1 over D squared. So 1 over 0.25 squared. And that will give you if you put it into your calculator, you get to use a calculator in science, yeah? So you just don't do it in your head, just do it in your calculator. Even if you know 2 + 2 is 4, use your calculator. Okay, 0.25 squared, and then you get 16. Okay, and that's how much more that the light intensity has increased by. Yeah, there you go. Good. Okay, so I want you guys to quickly try one. Let's see who is the smartest person in the world. Who's the smartest? Go on, go. First one to answer it. Here's the question. Go. First one to answer. I'm looking for the chat. I'm looking at the chat. 8. Oh, who is it? It was Sprays BTW. Well done, Sprays. You sprayed that one. Okay, good. So that's it, guys. Well done. And the answer was actually it was 6. Oh, you got it wrong though. You got it wrong. Oh, Sprays. You tricked me there. You got it wrong. Okay. So let me explain it one more time. Okay. So it was 32. It became 4. Yeah. So we're gonna let me just go back for this. Sprays, you tricked me, mate. So final distance was 4. That goes here. Initial distance was 32. That goes there. Okay. So it's gonna be 4 over 32, which is 0.125, and then you're gonna do 1 over that number squared, which is 64. There. Okay. So the intensity increases by 64 times. Okay, good. So anyway, that was the inverse square law. What other topics do you guys want me to go through quickly? Don't say triple content, because triple content is coming up straight after this, yeah? So nothing triple. What? Go on. Next one, let's see. Huh? Oil rig. You guys want oil rig? That oil rig is chemistry, my brother. Oil rig, that's chemistry. Metabolism, that's not coming up. Blah, blah, blah. Organization. Okay, that's all triple. Okay, we'll do xylem and phloem very quickly. Looks like everyone's asking for xylem and phloem, so yeah, let's do that. That's actually an important one. Good suggestion, whoever said that. So we're gonna do xylem and phloem, transpiration and translocation. Okay, let's go. And I think after that we'll have to call it a night. Those who said enzyme practical, we did that actually at the start of the live stream. Okay, so that was at the start of the live stream. Also, secondly, triple content is coming up straight after this. So binary fission, that's triple. The other one's triple as well. So we're going to come through all of that. Okay, guys? Here we go. Last bit, then combined science. We're going to have to call it a night. Get ready. 7 Kings, let me know if you want me to come down and test you guys out. But I think right now everyone's too busy revising. Okay, here we go. One second, iPad's loading, okay? One sec. Monoclonal antibodies, that is going to be triple science. So when we do triple science, we'll do monoclonal antibodies as well, yeah? So right now, we're just about to go through xylem, phloem, transpiration and translocation, okay? We're going to go through that very quickly, okay? Here we go. Get ready, everyone. And here we go. So here's a leaf, yeah? Here's a plant leaf. So we've got the top layer of the plant, the top layer and the bottom layer. It's called the epidermal tissue, yeah? Now, the difference is that the top layer has a waxy cuticle, okay? And the waxy cuticle is there to prevent water loss, yeah? The lower layer has special cells called, my bad, we'll come back to that in a second actually. So waxy cuticle at the top, the lower layer has guard cells, okay? And those guard cells work together to make a little hole at the bottom of the leaf called stomata. Yeah, that's the name of the thing. All right, moving on. Then we've got the upper layer here, underneath the epidermal, it's called the palisade mesophyll layer, the palisade. And what's special about this layer, we're coming to xylem and phloem in a second, yeah? What's special about this layer is that the cells are tall. They are very tall cells, so they can have a large surface area to absorb carbon dioxide, yeah? Okay, so what happens over here? The main reaction that happens here is photosynthesis. That's the main reaction that's going on here. Okay, moving on. Then we've got the next layer, which is called the spongy layer, or the spongy mesophyll. That's the one directly below the palisade. And over here, we've got loads of air spaces for gases to move in and out from. So the gases that go into the plant are carbon dioxide, okay, and the gases that come out of the plant are oxygen and water vapor. Yeah, so oxygen and water vapor both come out, and carbon dioxide goes in. All right, moving on. So let's talk about xylem and phloem. 1 second. That's stomata. We spoke about that earlier. Stomata can be open like this when there's a lot of water in the plant, but if the plant loses water, it will become closed. Yeah, that's the stomata. Good. So here we go. Xylem. Xylem vessel, very big one. Xylem and phloem work together. So we've got xylem transports water and minerals up the plant. Xylem only goes one way. So here's how you remember it. Xy, high. So xy goes high, but flow can go low. Yeah, that's a little gangster rap. So xylem only goes upwards, yeah? Phloem can go up and down. So it can go high, but it can also go low. So xylem is made of dead cells, yeah? It's dead cells. It has a hollow tube, and it's covered with lignin, yeah? So those are the three things for xylem. Dead cells, hollow, covered with lignin to make it strong and waterproof, and it carries water and minerals up the plant. That's xylem, okay? Phloem is used for translocation, yeah? So that means it moves sugars. Which way? Up and down. So it can move sugars up and down the plant. So how is the phloem structured? Number one, it's made of living cells, as opposed to xylem, which was made of dead cells, okay? It has these little things called sieve plates. And finally, it has something called a companion cell, which makes energy for the phloem. It makes energy using mitochondria. Done. Okay, final bit. Let's quickly look at the transpiration stream. How does water move through the plant? 4 points, and we're going to end it there for combined, yeah? Here we go. Number one, water evaporates out of the plant, and it diffuses out, okay? More water is then drawn up the xylem. Number three, this is called the transpiration stream. And finally, water enters the root by osmosis. There you go. That's how water goes through the plant. Smashed it. So guys, yeah. Okay. That was combined science. We're going to move on to triple science. Get ready, guys. Okay, combined students. Well done, guys. It was very good. So now look, here's what I want you to do, honestly. You see all this information that I just gave you? This all has gone into your brain, okay? What you need to do is you need to let your brain compartmentalize that and turn it into memory, so that when you wake up in the morning, you don't forget everything. So what, like, if you really want to do well, do yourself a favor, just have some food, go to sleep, wake up a little bit early tomorrow, watch this one more time, and go straight to your exam, okay? And secondly, do not speak to your friends before you go into the exam. They're just going to mess you up. They're going to make you feel stressed. And when you feel stressed, your brain turns off. You won't even know what your name is. You're, oh, what's my name? I'm too stressed. Do you understand that? Okay? So, cool. Well done, combined science. Triple science. Let's go. We're going to go through triple science now, guys. Get ready. Here we go.
Triple science asking, do they still need to know all of the combined?
Triple science. You still need to know all of the combined anyway. So, it's good that you guys came here, yeah? But anyway, we're going to go through triple science. The first one. Let's see if Anas from Loxford is here. Anas, if you're here, tell me, how do bacteria divide? Oh, Anas not here. Okay. Anyway, here we go. So, bacteria divide by a process called binary fission. Okay? Wow. Amazing. Triple science students are on it. They're just replying so quick. Okay, so they divide by binary fission, okay? And so prokaryotic cells, bacteria are prokaryotic, yeah? Which means they don't have a nucleus. They divide by binary fission, and they make multiple copies of themselves, yeah? Now, here's the thing. When they divide by binary fission, here's what happens. You have chromosomal DNA in the bacteria, and you've got these little rings of DNA called plasmids, okay? The chromosomal DNA doubles, but the plasmids can multiply many times, 4, 5, 6, 7, yeah? So that's the first thing that you have to say by binary fission. Chromosomal doubles, plasmids multiply many times. Then, when the cell divides, okay, you're going to have two daughter cells, but they're not going to be identical. Why? Because they have different number of plasmids. Does that make sense? So, you know how mitosis makes identical cells? Binary fission does not do that, okay? All right, moving on. Practical. How can we investigate how well an antibiotic kills bacteria? Here's what we're going to do. Step one. So basically we have, imagine this, we have all these different antibiotics, and we want to know which antibiotic is the strongest at killing bacteria. Yeah? Step 1, we're going to sterilize an inoculating loop, okay, by passing it over a Bunsen burner like this. So over there, it gets hot. Okay, now what you're going to do is you're going to let it cool down. Why? Because if it's hot and you put it straight into here, you're going to kill all the bacteria. So you're going to let it cool down, and then you're going to put it into the bacterial culture. Okay, so step 2, place the loop into the bacterial culture. Step 3 is once you grab some bacteria, you're going to spread it over the agar nutrients like this. You're going to spread it over that, nice and even. Then you're going to grab some paper discs, okay, and you're going to put those paper discs into different antibiotics using sterile forceps, yeah. And also one of them is going to be just water. So you put all those paper discs into different antibiotic solution. Take them out and put those paper discs and the water disc onto the agar jelly that you put the bacteria on. Then you're going to put a lid on it, okay, and you're going to cross tape it. But you're going to cross tape it like this, like cross tape, yeah, not circle, cross tape. Why do you want to do cross tape? Because you want to allow oxygen to still be able to enter from the side and get to the bacteria, okay, and to prevent anaerobic bacteria from growing. Then invert to stop condensation, incubate, so you leave it at room temperature, and finally leave it for a certain amount of time. So that's what you're going to do. So you're going to flip it over, you're going to leave it there, come back maybe in a day or two. When you come back and you open the lid, you're going to see something very special. You're going to see little areas where the bacteria did not grow. So I don't know if you can see it very clearly in this diagram, but can you see around this pink one? You have these clear areas. Those are called inhibition zones. And you've got one over here as well. And then what you're going to do is you're going to compare the size of those zones. So for example, you're going to measure it, yeah? The biggest one means it killed the most bacteria, therefore it is the best antibiotic. Okay, so look, that's the biggest. Let's give that number 1. That's number 2, and that one's very small, number 3. Now, should you expect an inhibition zone around the water one? Should you expect one? Should there be an inhibition zone around the water paper disc? No. Yeah, should not be. If there is, then that means your experiment is faulty. Okay, there's a problem. Okay, so the water is a control. It should not have an inhibition zone, and so that means that you've done a valid experiment. Okay, now how do you make sure that your results are reliable? You have to keep the following things constant. Yeah, what did we change? What was who can tell me what was the independent variable? It was the different antibiotics, right? What was what was the dependent variable? It was the size of the inhibition zone. Okay, so you changed the antibiotic, and you saw the size of the inhibition zone. Now, what do you keep the controlled? What do you keep the same? The following: 1, 2, 3, 4. Okay, so the concentration of antibiotic, the size of the paper discs. Make sure the whole thing's sterile. There's and also no overlapping. There, done. Okay, next topic. Get ready, triple science. Get ready. Papa. Oh.
If they ask us for the area, can we figure out the area of inhibition, this or just this?
The area of inhibition, yeah, from the middle of the circle. Okay, get ready. We're gonna go through monoclonal antibodies and the pregnancy test kit. Okay. ExamQA, please can you come to St. Augustine's? How do you say Augustine? Yeah. All right. Come. We're gonna. If there's time, I'll definitely come to your school. Okay. But make sure you get it right. Otherwise, your friends will not respect you. Cool. Get ready. Tom Webster is a op. Get ready, guys. 1 second. It's loading. It's loading. Okay. So we're gonna. Q boys. Oh, I still haven't come. I can't believe it. Q boys. We're coming Q boys soon. Okay. So now, anyway, we're gonna quickly go through monoclonal antibodies. Then after that, we're gonna go through the pregnancy test kit, and then after that, we're going to say goodnight. Actually, we'll do a Q&A and then we'll say goodnight. Okay. I don't know why Don't bring that up, yeah? Don't bring up the handstand. Oh. Okay, guys, sorry, 1 second. My thing's loading, yeah? Give me like 3 seconds. I know everyone hates bio, but to be honest, bio, I think, is the easiest to learn, but it's the hardest and when it comes to questions. Anyway, too much talking. Let's get straight to monoclonal antibodies. Here we go. So, first of all, you have to know this, yeah? B lymphocytes, okay, are a type of white blood cell. That's a B lymphocyte, okay? Now, these B lymphocytes, they produce antibodies. So, antibodies are proteins that have a specific shape and they bind to the surface of pathogens. Those surfaces are called antigens, okay? So, antibodies bind to antigens. Anyway, we know that antibodies can have all different types of shapes, okay, but we don't care about those ones. We want antibodies that all have the same shape, yeah? So if you got a group of antibodies that have the same shape, they have a special name. It's called monoclonal antibodies, yeah? So just to quickly summarize, B lymphocyte makes antibodies. Antibodies bind to antigens, and if you have a group of antibodies that all look the same, they are called monoclonal antibodies, yeah? Done. Okay, now, how do you create monoclonal antibodies? First of all, you're going to take a mouse and you're going to inject it with an antigen. Step 2, this will activate the B lymphocytes inside that mouse. Okay? Step 3, we're going to extract the B lymphocytes from the mouse. Okay? And then step 4, we're going to get a tumor cell, which is a cancer cell. And we're going to fuse the B lymphocyte and the tumor cell together to form a hybridoma. Okay? And once you've done that Dragon Ball Z fusion, then you're going to take the hybridoma. Now, what's so good about this hybridoma? What's so good about it? It has the properties of both of them. Number 1, it has the properties of the B lymphocyte because it can make antibodies, and it has the property of the tumor cell because it's rapidly dividing. Okay? Then, once you got all your hybridomas, you allow them to replicate, and then you extract all the antibodies out of the hybridomas. Okay? So you grow them and you extract it. Done. There you go. That's how you make a bunch of monoclonal antibodies. Okay? Now we're going to go to the pregnancy test kit. Someone said, give a summary of this. I don't have a summary for this particular one, but the steps are at the bottom, okay? So when you come back to this part of the video, when you watch it later, you can just go through it one time, okay? Let's go. So we're going to quickly talk about, wait, let me skip this. Here we go, pregnancy test, okay? So this is called a pregnancy test strip. What do we use it for? What do we use a pregnancy test strip for? Who can tell me what do we use it for? Okay, give wrong answers only. Wrong answers only. What do we use a pregnancy test strip for? Okay, so step 1. On one side, we have antibodies that are that have these blue beads attached to them. Okay. Step 2. Oh, it comes off too funny. Okay. So on one side of the strip, you have antibodies. Okay, and obviously they all have the same shape. Yeah, and they have blue beads attached to them. Okay. Then on the other side, you have antibodies that are the same as the other one, but these are stuck onto the strip. Okay, so they can't move. The ones on the left, they can move, but the ones on the right are stuck there. Okay, and just to quickly make sure you understand, these antibodies and these antibodies have the same shape. Okay, the only difference is the ones on the left have a blue bead on them as well. Yeah. Okay, so the pregnancy test strip is used to find out if you're pregnant. Yeah. So when the lady urinates, if she's pregnant, her urine will have a hormone called HCG if she's pregnant. Okay, that hormone is complementary to the antibodies on the strip. Yeah, so the lady is going to urinate on the left side where the blue beads are. Okay, so suppose she's pregnant. That means the hormone will attach to the antibody and the blue bead. So together, that whole hormone, antibody, blue bead complex, all of that, okay, is now going to move down the strip. As it moves down the strip, these antibodies on the right are ready to catch that hormone. Okay, so they're gonna catch it. When they catch it, that whole combination, antibody, hormone, next antibody, blue bead, all of that is gonna stay in one area, and that area is gonna go blue. Okay, that's how you know she's pregnant. Now, if she's not pregnant, if the lady's not pregnant, when she urinates, there's not going to be any hormone present. So the whole combination will keep going. But what do you think is going to happen now? What do you think is going to happen to this? When it gets to this area, do you think it will attach to the antibodies or not? Catch me, mate. Is it going to attach or not? No, it's not. Okay? It's not going to attach. Why? Because the hormone was not there, and therefore the whole thing is going to remain clear. It's not going to become blue. Done. That's it. Okay, everyone? So it will not turn blue. Good. All right, that's it. So that was the triple content as well. So triple, obviously, everything that comes in combined comes for you guys as well. And you had some extra stuff there. Okay, so that was it. If anyone has questions, type it in the chat. What do you want to say? Alright, fine. Alright, calm, hard. So guys, triple content. Zishan's gonna speak to you again while I go bang out 100 press ups. Yeah, count them back.
Ask questions. Hammer's gonna answer the questions. He's gonna do some press ups just to get ready to answer your questions. Look. He's doing press ups just to get ready to answer some questions. Yeah. You see him there? I'll keep him there. In fact, I'll keep him there. Okay, give me 1 sec. Actually, sorry, my bad. Okay, guys, so that's bio done, but stay just for the Q&A. You know, like Mohammed said, the advice is given you. Go over the video again. We're going to put timestamps on everything, so you know you can watch it again, take more comprehensive notes. You know, have a sit down and watch it properly, so that you know you can really digest all the information. Just to reiterate, exclusive code just for the live stream lot. We only do these live streams once a year, so we thought, you know what, we might as well make a special code for you guys. For the 2024 exam season package only, normally it's £39 and you get all the content. Just for the live streams, we're doing a £10 off offer. Go to I have read and agreed to the website terms. Then go to coupon code and you write live stream. This is exclusive code just for you guys. Okay, click apply and then that's it. You get your £10 off. Okay, and you get so much content you would not believe it. Look at this. Look at this content. Exclusive viewing. I'm showing you guys. And this guy just done some press ups to get ready for your Q&A that he's definitely going to do, of course. But now look at this. Physics paper 1 and 2. You haven't even started that yet. So think about all these slides and resources that you can utilize whilst the exam is not done yet. Okay, have a look at this. All these slides, right? I'm going to go to wave basics. Okay, I'm going to go to AQA physics notes. Okay, in the morning of the exam, we have notes already written out for you, you know, very comprehensively, right? If I was you on the morning of the exam, look at this. Look how beautiful this is. If I was you on the morning of the exam, I would print this out, read this on the way to school, you know, have a quick, you know, recap of all the different things I've learned. You know, very nice, beautiful notes made by our team, all original notes. You know, they're very amazing, if I don't say so myself. And of course, you got the slides, all the slides that we've made personally by Mohammed, right? Transverse, longitudinal, what else we got? I want to show you one of my favorites, one of my personal favorites. We got lenses here, right? Because I wear glasses, so I had to learn about why do I need to wear glasses, right? I was very curious. And Mohammed made this slide just for me. Okay, look at this, beautiful, fantastic.
Well, apparently I did. You guys saying 79, bro? I did
79 press-ups.
Nah, they're waffling.
Okay.
I did 100. You guys can't count.
Now, all of this content you're gonna have for yourself. What is our speciality? What is our, you know, USP? Our USP is that you can pretty much learn the entire spec in a matter of a few days. Okay. look, you see in the TikTok videos, Mohammed goes to East London, Northwest London, South London. He tests these finished kids. Again, apologies, they're very nice kids. I've met them myself, right? Very nice boys. But they go from zero, being able to answer 0 questions, a quick 2-minute, explanation from Mohammed, and now they can answer very, very difficult concepts. Watch the videos, go back, look it for yourself, right? And these slides are available here just for you. You can't get it anywhere else. I'm going to show you one more thing. Boom. All the specs you got here as well. AQA, EdExcel, OCRA, OCRB, Chem, Bio, Physics. It's all there for you on a plate. You just got to eat the plate. Sorry, you just got to eat it with a nice spoon. We've made the spoon for you. We've collected all the food for you. You just have to digest it and eat it out nicely. Okay. Let me show you one more thing.
I'll just say while you're loading that up, we just posted Beale School on TikTok.
Check that out.
We just posted it. Now look, Southgate have 700,000 views. Okay, whoever puts comment your school, whichever school gets the most like when they come to that school next. When is it? Monday. On Monday. Okay. So go to our TikTok, the latest video from Beale School. Put your school in that. Put it in the comment. We're gonna come to that school next, and we need to make sure that we smash up. Come on, Bill, you guys are doing well. Okay, sure. Is it time for Q&A?
Okay, so look, all of these slides just for you. Just for you. If I had these in my GCSE times, do you know what I mean? I wouldn't be able to believe it. I would pay double. I'd pay double. Right? But, because we understand that it's exam season now and people are desperate, therefore we made the discount just for you guys. Code livestream. Let me put it on the chat for you guys. Let me put it in the chat. Let me put it on here. Code livestream. Code livestream. Code livestream. Of course, don't stay up too late though, because you need to go to bed. But I would recommend purchasing this before you sleep, so that at least in the morning you can still utilize these slides that Mohammed used. All the slides that he used today are available on here, and that's all from me. Again, subscribe for paper 2s bio, and of course the paper 1s and 2s for physics and chem. We're going to be doing this every single exam night before, where we just cover all the predicted topics so that you guys can smash that exam. Thank you very much and have a good night.
All right, Zishan. By the way, Zishan's going to be teaching guys paper 1 maths, yeah? So when's paper 1 maths?
Next Thursday.
Thursday, Zishan is gonna go for paper 1 maths with you guys, okay? So get ready for that, guys, yeah? Again, always try to do better on the paper 1s, because that way you can what's it called? Anyway, so let's go through some more topics then. For whoever's still here, tell me what topics you want to go through, guys. I don't mind. Let's do it. We'll do like another, let's say until 10:30. Yeah. Let's say until 10:30. Until 10:30. Okay. We're gonna go through more topics. So go. What's next? Let's have a look. Menstrual cycle. Okay. By the way, just to let you know, the topics that we'll go through, because you guys do different exam boards right now. Okay. We've done everything from AQA exam board, but again, if there are topics you guys want to go through, we'll go through it now. Okay, go. Let's have a look. So we've got kidneys. Kidneys is going to be paper 2 for AQA, but if you want to do kidneys, we'll do that. Developing drugs. Metabolism. We've done transpiration translocation. Protein synthesis. Alright, let's do that. Okay, fine. We'll do, what is it? We've done osmosis practical already. Okay, we'll do developing drugs, yeah? We'll do developing drugs, because I think everyone's talking about that right now, developing drugs. So that'll be a quick one, okay? Developing drugs. What was it, B2? One second, guys. Get ready for that. Wait. Drug development. Okay, actually that is a big topic. Here we go. Drug development. Okay. Coming up now. Right. Okay, guys. So in drug development, we have 3 stages. Yeah. Now, first of all, let me just tell you something. Okay. You see the drugs that you have in the supermarket or in the pharmacies. Those medicines, those drugs, have to be developed in order for them to be safe for us to consume. Okay. So in order for that to happen, it has to go through 3 stages of drug development. Stage one is testing on cells and organs. That's stage 1. Yeah. Now, why do we test on cells and organs? Because you want to see if the medicine works, pretty much. Yeah. The problem with this stage is it does not work for all medicines, and it does not work on drugs that have multiple body systems. For example, if a drug is trying to affect like the heart, the kidneys at the same time, it's not going to work very well. Yeah. So just to summarize, stage 1 on cells and organs. Stage 2 is on small animals. Yeah, like animals like rabbits, mice. Why do we use them? Because number 1, they're small, and number 2, they are cheap. Yeah. Then, what's the point of this stage? The point of this stage, 2 words: efficacy and toxicity. Those are the 2 main reasons of this stage. Efficacy means to see if the drug's actually working. Is it curing the headache? Is it lowering the blood pressure? And toxicity to see if it's dangerous to the cells. Yeah. Once it passes stage 2, we go to stage. Okay. The negative thing about stage 2 is ethical issues. So sometimes what happens is people will say, oh, it's not fair, why are you testing on an animal, the animal has no right, etc. So there's pros and cons. Yeah. What's the pro? You test the drug, and you get to help people through testing on an animal. But the disadvantage is that testing on animal is not fair because it harms the animal. Step 3, now that's when you move on to human beings. So whenever you move on to human beings, we have clinical trials. Okay. So clinical trials, we first start on healthy humans. Okay, guys, starting on healthy humans. Okay. So why do we do healthy humans? Because if there is a side effect, they're less likely to suffer from it. Yeah. And the main point of this is to check for side effects and check for optimum dose. Yeah. That's the 2 benefits of clinical trials. Moving on. Within this part, we have something called a placebo. What's a placebo? A placebo is a sugar pill. It does not contain the drug. It looks like the drug, but it doesn't contain the drug. Yeah? Okay. Now, what's a blind trial? In a blind trial, the doctor knows what he's given to the patient, but the patient doesn't know. So the patient doesn't know if they're getting the placebo or if they're getting the real drug. What's the point of this? Why are we not telling the patient? Because we want to remove something called psychological placebo effect. Imagine someone's got a headache, yeah? And you give them a pill and you say this will treat your headache. But deep inside you know that you just gave them a water pill or a sugar pill, and they say to you, oh yeah, I feel much better, thank you so much. You know that they are waffling. Why? Because you didn't give them the drug. Does that make sense? That's called the placebo effect. So a blind trial, the doctor knows, the patient doesn't know. And the benefit of it is what? Is to eliminate a placebo effect. Yeah. Now we have another one called the double blind trial, where neither the doctor nor the patient knows who's getting what. And if they ask you what's the point of that, you say to prevent bias. Done. Okay. That's drug development. Cool. Next topic. Come on, guys. Let's do one more. We still have 6 minutes. Let's do one more topic. Yeah. Next topic. I was nothing. Next topic. What is it? Menstrual cycle. Okay, menstrual cycle doesn't come up in paper 1 for AQA. I'll go through it, but just to let you know, if you're in AQA, menstrual cycle is not coming up for you in paper 1. But if you still want to learn it, then of course, by all means, we can go through it quickly. Yeah. But just to let you know, it's not coming up for paper 1 in AQA. All right, guys. Here we go. Anyway, it'll be three minutes. Easy. Here we go. The menstrual cycle. There are 4 hormones in the menstrual cycle that you need to know. The first one is FSH. F for FSH, F for first. Yeah? The second one is estrogen. The third one is LH, and the fourth one is progesterone. Okay, these are the 4 hormones, and that's the order. So F for first, then it's estrogen, then it's LH, and then it's progesterone. Yeah. All right, calm. I just want to get loud. So step 1, FSH is produced by the pituitary gland. It comes down to this area, which is called the ovaries. It goes there. Okay. Step 2, when it gets there, it causes a follicle, which is a little egg, to mature. That's what it does to the follicle. At the same time, it also stimulates the ovaries to produce estrogen. Okay. Step 4, estrogen travels to the pituitary gland. Okay, it travels there, and it tells it to stop making FSH and instead to produce LH. So just to quickly recap what's going on right now. Pituitary gland, FSH comes down to the ovaries. Estrogen goes back up to the pituitary gland. It says stop the FSH, send down now the LH. Yeah? Okay. At the same time, estrogen also, so LH is coming down, yeah? LH, we'll talk about LH in a second. But at the same time, estrogen also causes the lining of the uterus to grow. Alright? Moving on. So when LH comes down from the pituitary gland to the ovaries, when it comes down, okay, it causes an egg to be released. So an egg gets released from the ovaries into the fallopian tube. That's what LH. So LH literally lets the egg loose. L for LH, L for loose. Okay? Now, progesterone, what does that do? That's the 4th hormone. Yeah? Progesterone maintains the lining of the uterus. That's it. That's all it does. Okay? So, again, just to let you guys know, this topic, not paper 2 for AQA, but Edexcel OCR, do this in paper 1. That's why I'm doing it. But for paper 2 students, so for AQA students, you guys don't need to know this for paper 2, yeah? Alright, good. So that's it from me. Zisha's gonna say bye to you guys. He's gonna wish you guys a good night. Rest well, eat well. Watch this tomorrow. We're gonna put the timestamps for you as well. Okay? Bang.
So we're gonna edit this video, put all the timestamps on it, so that you can watch it again after, and take more sort of slower notes. Like I said, guys, limited time only. After 12 midnight, the code is not gonna be there anymore. The code is not gonna be there anymore after midnight. Okay? So use this code wisely. Use it tonight, right? We're going to disable it because, again, this is exclusive code just for you guys. What do you get with the code? Look, I showed you guys the content. Come on, guys, let's be real. Chem, bio, physics, English, maths, right? There's too much stuff. There's too much stuff there, and it's not even a thing where, you know, it's going to overwhelm you. You know, the topics that Mohammed went over today. For example, in biology, I would get those specific slides. If I was you, and then I'll just go over them again. The topics that he didn't go over, don't go over those slides yet. Wait for paper 2 to come up. Okay, paper 1, go over those slides, etc., etc. Be wise with your revision. You don't want to overwhelm yourself. You don't want to learn stuff that's not even going to come up. Hence why we've done this live stream, right? This predicted topic live stream. So use code livestream, you know, go to the checkout, you know, write the code in the coupon area, and you get this exclusive discount. Again, like I mentioned, the code is going to be, we're going to remove the code after midnight, so use it tonight. Ask your parents if they can kindly support you with your learning, and you know, all these slides will be available just to you. Again, the slides are very good. In the morning of the exam though, I would probably recommend just going over the notes rather than an entire slide, just because it condenses. So for example, if I go back here, for example, did you go over ecology? You didn't today?
No, that's paper 2.
That was paper 2. So for example, if I go to biology right now, and I go to cell biology is paper 1, right?
Yeah.
Yeah. So I might go over the slides tonight, but tomorrow morning, I'll print out these notes, right? These ExamQA notes, and I just look over them instead, okay? Because it basically summarizes the practicals, the content, the different techniques, things you need to memorize. It summarizes it all for you very nicely, very condensely, very briefly, okay? So you be wise with your revision, how you use the platform, using the code livestream, and there's quizzes there as well if you want quick fire stuff. But be wise, don't overwhelm yourself. Don't do an entire worksheet, for example, right now. Maybe looking at mark schemes is useful, but I think writing and printing out worksheets, for example, and going over them for the entire night, if that's your plan from the get-go, fair enough. But I'm talking about closer towards the exam, when there's only a few hours left to spare. Maybe just look over the notes and try to consolidate all the information. These slides, they're not available anywhere else. Mohammed has, hand-made them. He's handmade them. He's an artist. I might get him to draw a picture for you next time. Request a sort of image, and he will make that for you. Very, very amazing slides here for you guys, right? In fact, let me show you one of my favorite ones. Like I showed you earlier. We got it's one about chemistry of the atmosphere. That was a very nice one. Yeah. Who's this man? C9. Oh, it's Mohammed. C9. Let me show you what these ones, right? These will blow your mind. I won't lie, these will blow your mind. The comments. Click the paint file. Click the paint file on the pass thing. Do you know what? I need to explain some of them to you though.
You guys were mad with the comments.
Mohammed was very interested by the comments, and he actually low-key wanted more, but he didn't want to say it because that's all the specs for you here: AQA, EdXL, OCR-A, OCR-B. We've got all of them there for you. English and maths as well. AQA, EdXL, OCR. I'm going to show you one of the slides that are very, very nice, very lovely, very colourful. And make sure you subscribe. Make sure you follow us on TikTok so you can get all the announcements for when we do these lives. We appreciate the link, you sending the link to all of us, your friends. Sorry, spread the link to your friends. Don't gatekeep. It might seem like all these. If I send the link to everyone, I'm going to lower the grade boundaries. I'm going to gatekeep so I can lower the grade boundaries. But if you think about it, it doesn't really make sense. Like, what's a few thousand people going to see the live stream going to do, right? Think about how many millions of students are actually taking the exam. 1,000-2,000 people. It's not gonna make a difference in reality. I need to do timestamps. Oh. First time.